Okay, so I think we'll go ahead and get started. Welcome everybody to the April iOS seminar on spatial patterns of speciation. Before we get started, I'll just let you know this is the last seminar until we get started again in September. And we'd really like to get ideas from the community about different topics that you'd like to hear seminars about. So we'll send an email out after the talk to try and get some suggestions from everybody. Okay, and with that, it's my pleasure to introduce Gideon Bradburd, who is going to tell us about the geography of evolution. And so Gideon started his career with a bachelor's degree in ecology and evolution at Yale, which he completed in 2008. He then started a PhD at the University of California, Davis, supervised by Graham Coop and Brad Schaefer, which he finished in 2015. He then had a short postdoc at UC Berkeley with Michael Nachman and Erica Rosenblum. And he's currently assistant professor at the University of Michigan in the Department of Ecology and Evolutionary Biology. And research in Gideon's group focuses on understanding the geography of evolution. And people in his group combine empirical data analysis with approaches in statistics, computation, and theory to learn about the processes that drive and maintain complex spatial patterns of genetic information. This includes the development and implementation of new population genetic models and analytical methods for describing population structure and admixture, as well as studying local adaptation, coevolution, and natural selection. And of course, all of these are relevant to understanding speciation. And so with that, I'd just like to hand over to Gideon, who can tell us about spatial patterns of speciation. Thanks, Gideon.
Well, thank you. I'm just trying to fix my screen here. Thanks so much for the invitation. It's really exciting to get a chance to talk to all of you. And thanks so much also for the nice and well-researched introduction, Sean. So yeah, can folks see the screen okay? Okay, great. Well, as Sean mentioned, my work, the work in my lab is focused on spatial population genetics, or the geography of evolution more broadly. And I was really excited to get the invitation to speak today because the study of speciation intersects in really fun ways with the spatial questions that my lab works on. And in particular, one of the ways that I think it intersects the most, or one of the ways that I think about the most, is about the role of geography in speciation. So I'm going to very briefly walk you through what I think about the role of geography in speciation, and presumably this is things that you've gotten lots of talks about in the past. But I just want to share sort of the papers that have informed my thinking as I developed as a researcher. So, one of the first things, right, is that closely related sympatric species are uncommon. In general, this is a great figure from Barraclaw and Vogler 2000, showing that the relationship between, if you're plotting the relationship between genetic divergence or phylogenetic divergence on the x-axis and the degree of symmetry on the y-axis, you tend not to see many points in this upper left-hand corner of closely related, fully sympatric species. They exist, but they're certainly rare. And when we see them, this is going back to Pointinor 89, we see that they have elevated rates of prezygotic reproductive isolation compared to similarly genetically diverged allopatric taxa. So you see a lot more points in this upper left-hand quadrant of small amounts of genetic differentiation with lots of prezygotic isolation in symmetry compared to allopatry. So combining these two pieces of information, I think that most, and yeah, I guess I think, and maybe many of you agree with me, that most speciation is probably occurring in allopatry. And this is another Barraclaw classic from Yale Kiesel and Tim Barraclaw in 2010. This is a paper I really love, fun to think about, it launches a lot of questions in my head. So this is looking at the probability of in situ speciation on islands of different sizes, and island sizes on the x-axis, and the probability of speciation shown on the y-axis for these different taxonomic groups. And what I find really interesting is that although in general you see higher rates of speciation happening in situ on larger islands, presumably reflecting something about the opportunity or prevalence of allopatry. The rate at which that happens varies. And that's summarized really beautifully in this graph with these hand-drawn little adorable figures showing the relationship between the geographic extent of the smallest island on which they observe or they infer in situ speciation to have occurred, and the mean FST at this sort of small-ish or medium-small spatial scale in that taxonomic group. So you see that snails and flowering plants have a lot of genetic divergence over the spatial scale compared to bats and butterflies, and then that is predictive of the size of the island that they can speciate on. And in general, things that have shorter dispersal capacity can speciate on smaller islands, which kind of makes sense. And presumably this is reflecting what counts as allopatry for these species, or the probability of getting into allopatry. And so the big take home of all of these points is that if we're thinking about the geography of speciation, or how speciation happens in space more generally, we really have to think about dispersal, a species' capacity for dispersal. And we can learn about dispersal from studying spatial patterns of genetic variation. That's a lot of what my lab is focused on. So when we talk about spatial patterns of genetic variation, we're usually thinking about isolation by distance. And PopGen is confusing because there's also identity by descent, and sometimes people study identity by descent with irritable bowel disease. So there's lots of IBDs to stack up here, but this is isolation by distance, which I think is best summarized by Waldo Tobler's first law of geography, which is that everything is related to everything else, but near things are more related than distant things. And I want to just walk through how I think about how that arises, because it informs sort of the work that the lab is doing. So if you imagine, right, here's me, this little dot in the present, on some spatial landscape. Can you all see my mouse? Okay, yeah, so on some spatial landscape in 2D space, and there's lots of other individuals who are these little dots that I co-occur with. I have 2 biological parents and 4 biological grandparents, et cetera. And if we assume sort of 2 simple rules, one of which is that dispersal from anyone's birth location is distance limited, and second, that mating opportunities are also distance limited, so you can't mate with anyone anywhere, then we observe this pattern where the sort of spatial cloud of my ancestry grows as I go back in time. And one way to think about that is that your pedigree is spatially autocorrelated. And so now if I add you to the picture, right, I'm picking out your pedigree in geographic space. And in this case, it looks like we have to wait 8 or 9 generations before we have the first pedigree overlap between us. So that's this, kind of hard to make out, but this little line right here is an individual that shows up in your pedigree and my pedigree. And so if we both inherit genome from that individual, we'll be genetically related, identical by descent, the other IBD, and how long ago back in time that we share this ancestor controls how much genome we co-inherit, and therefore how closely related we are. And so now if I add in a third individual, Joe, or whatever, Joe lives close to you in the present. And as a result, your pedigree and Joe's pedigree overlap much more quickly in geographic space, but then also the pedigrees actually merge much more quickly as well. And as a result, you're more closely related to Joe than either of you is to me, right? And so this gives rise to a pattern of isolation by distance where proximity predicts relatedness by determining the timescale over which you start to see these pedigrees merge. So I'm going to talk to you about a project that's sort of very fresh, just in the past couple of weeks out of my lab. It's still unpublished, looking at how to learn about rates of dispersal, these rates of the sort of spread of pedigree or of genetic ancestry across space through time, using trees. And this is work that's led by my fabulous postdoc, Mike Grundler, and is in collaboration with Jonathan Terhorst, who's in the Department of Statistics here at University of Michigan. So learning about dispersal, focusing on trees in space. And the tool that we're using to learn about dispersal is the ancestral recombination graph, for the ARG. which is a record of all of the coalescence and recombination events since the divergence of the sequences under study. And that ARG, or an ARG for a set of samples, specifies a complete genealogy at every genomic position in the data set you have. And just a quick shameless plug for my grad student Alex's review paper, we kind of wanted this to be called like everything you ever wanted to know about ARGs, but we're afraid to ask. It's the paper that we wished we could have read when we started reading about ARGs. So I encourage you to check it out if ARGs are something you're interested in. It's out now on PLOS Genetics. But briefly, you can think about the ARG as a graph that's embedded within the pedigree that describes the full record, as I said, of coalescence and recombination for a set of sequences shown here, A, B, C, and D, that are under study. And it can be thought of as a tree sequence or a sequence of local trees, here color-coded by genomic position that they each apply to. And so this is showing the record of coalescence between samples A through D within each genomic position. And those local trees are highly autocorrelated. And so you can see when you move from one genome position to another, in general, the topology is very similar, or the branch lengths are very similar, although it can change because of recombination events. So essentially what we're doing is like old school mitochondrial phylogeography, but instead of using a single topology or genealogy with a set of node heights, we can do this type of geographic inference that I'm about to talk about using many, many, many trees, this full distribution of local trees across the entire succinct tree sequence, which is a sort of a summary of the ARG. I'm not going to talk more about ARGs, but you can think about them as essentially as this set of local trees where some nodes show up across multiple local trees. And so there's therefore ways to share information across them. And I'll talk more about that. So we consider a data set that consists of this full set of dated local genealogies. And each node in this genealogy represents a real genome in a real ancestor that was shared by the set of samples in the modern day, which are 0, 1, and 2. And the only thing that we have to know here is that set of genealogies and their node heights, as well as the geographic positions of individuals in the modern day in XY space. So that's shown here on this little map. These are their geographic locations where we've sampled them. And notice that node 4 here shows up across multiple local trees. That's a shared ancestor from whom these individuals inherited genome at multiple places across the genome, which is really common, right? We inherit genome from shared ancestors in multiple places. And the approach that we take is sort of a really straightforward and simple, verging on stupid one, which is just to assume that we can model the flow of ancestry across space using parsimony. So what we can do is, and when I say we, I mean Mike Grundler, who is very much the brains behind this operation and a really fabulous scientist to work with. But Mike figured out a fast and flexible way to describe migration cost surfaces that determine or that record the total amount of distance that has to be traveled given different guesses at where these internal nodes are. And then we can very quickly place nodes at the minimum of those surfaces to get the most parsimonious dispersal history. So that's what I'm showing here. Each of these surfaces is, you can think about it, it's sort of analogous to a likelihood surface. It's a parsimony score surface. And we're placing nodes at the locations that have that lead to the most parsimonious migration dispersal histories, these minimum cost dispersal surfaces. And you can see also how information is shared about the location of nodes that show up across multiple independent local trees, right? This node 4 shows up multiple times. And as a result, there's information on where 4 occurred from both the locations of 0, 1, and 2, as well as the inferred locations of nodes 3, 6, and 5. And so in this way, we're able to leverage a really large amount of information or a large amount of data across the whole genome. So that's roughly how it works, or that's briefly how it works. We tested it using a variety of different spatial simulations, and it appears to work reasonably well. As we go deeper in time, the accuracy of our ability to infer the locations of genetic ancestors starts to decrease. But it's surprisingly pretty far back in time that happens. So I'll show you some applications to human data, and then I'll sort of end with a discussion of how I think this circles back to speciation research. So here's a single tree in space. This is the local tree for this genomic interval on chromosome 18 between 591,000 and 592,000 base pairs. That's the genomic region where we infer this genealogy. And this is each large circle here is a geographic location where we have data for sequenced humans, and circle size is proportional to the number of humans we see or we've sampled there, or that have been sampled there. And then the color, what we're doing is projecting this topology onto two-dimensional space and coloring each edge by the age of the edge. So more recent edges in this local tree are this sort of peachy beigey color, and older ones are this sort of dark red color. And you can see, there's this interesting sort of octopus spreading its dark red arms out of Northeast Africa. And those are some of the oldest branches, and then lots and lots of really recent connections between all humans in the sample, which are shown in this sort of peachy color. So we can do this for one gene. We can do this for all the genes. And so this is a summary of what we see across all of chromosome 18. And this is really exciting when we saw this result, because we know that humans came out of Africa. We know sort of roughly when that happened. And so it was really nice confirmation to see that a lot of these older branches are coming out of Africa, as well as highlighting, right? You can see the sort of background of this peachy beige color showing how closely related all humans are to each other, right? We share ancestry in the very, very recent past. And again, what we're doing here is just inferring, somewhat poetically, you can think about it as your genome existing at every point in the past across all of the ancestors from whom you will eventually inherit it. And what we're doing is just tracking not those locations, because we can't know about everyone's ancestors or all of a single individual's ancestors, but instead we're learning about all of the shared ancestors, and we're trying to identify where those individuals lived. And once we have that, we're just projecting that georeferenced tree onto this two-dimensional space to get this plot. Here's what it looks like for a single individual. So this is a modern day individual sampled somewhere in Northern Europe, maybe Norway. For the folks joining from Europe, I'm sorry if I got that wrong. And you can see, right, this line is showing the mean or the median path of the average ancestor. So this is just sort of plotting the centroid of where this individual inherited their genome from through time. And you can see in the recent past, right, that's somewhere in Europe. As we go farther back in time, it goes through the Middle East and into Northeast Africa. And that's the average location of where this individual's genome, or the bits of this individual genome that we can track, were at that time. Some other exciting things that we can do with this are to draw lines in space and then measure how genome that ends up in the modern day sample moves across those lines through time. And because we have this temporal component, we can pick up not just the average dispersal across some barrier or putative barrier or migration corridor through all of time, but the specific events at which we see lots of genome moving across those lines through time. So here we've drawn a line along the sort of northeast horn of Africa into the Middle East, and we can track how much of the genome that ends up in the modern day sample is leaving Africa in these different temporal windows through time. And you can see we infer there to be sort of two big spikes a little over 100,000 years ago. I'm not like the most confident in the world about the exact timing, because this really relies on us getting accurate node heights from this inferred ARG. And so I wouldn't be shocked if I wouldn't bet a ton of money on the exact dates, but I am really excited about the application to be able to infer the specific timing of dispersal events across possible barriers or possible corridors of migration. And I'll circle back to this. Some other things that I'm really excited about are that this allows us to define a sort of spatially explicit version of ancestry, right? When you do 23andMe or Ancestry.com and it says, oh, you know, Liz Scordato, you're 40% Irish or whatever, it says that is implicitly tethered to a point in time, right? And usually because this is, you know, targeted at American audiences, it's something in like the late mid 1800s. And that what it's actually saying is that you have a lot of, you know, third or fourth cousins who live in Ireland now. But what we can do, right, is say, hey, Liz Scordato, I've drawn this circle in space, and I'm going to try to quantify how much of your genome came from individuals that we infer to have lived inside that circle at a specific point in time. So that's what we're doing here, dividing the sampling area into sort of 4 different broadly continental regions. We can infer where those individuals are inheriting their genomes from backward in time, 10,000 years, 100,000 years, going back 500,000 years. And what's neat here, because it's humans, right, is that once we get back to 500,000 years, basically everyone's inheriting their genome from the same spot because we all have the identical spatiotemporal ancestry at that time period. And another way to look at that is shown here. So this is kind of a structure style plot, but the x-axis here is time. And what we're doing is tracking at the modern day, ancestry is just wherever you were sampled. And as we go back in time, it's where we infer your genome to have been found from. And you can see as we go farther back in time, all humans are inheriting their genome from the same spot, which is North Africa in the Middle East. And sort of we can build on that definition of spatiotemporal ancestry to think about ancestry flux. So this is showing, this is a map showing the direction of greatest migration that connects ancestral samples to modern day individuals through time. And so you can think about this sort of as like the average flux, the average flow of ancestry across space through time, and we can summarize it really nicely. And I think this is a cool summary of how genomes are ending up in modern day individuals. So this is an application to humans, which aren't particularly interesting from a speciation perspective, but for which we do have these really rich data resources. And in some ways, I think humans are sort of where the rest of us are all going, right, with developing data sets for either non-model systems or systems where there's more exciting or interesting natural history, or specifically for this, interesting speciation questions to ask. So some of the things that I think this type of approach could be really neat for is to learn about dispersal, both the average rate of dispersal, so we can estimate that by using a summary of the mean parent or parental node, offspring node displacement that we infer, divided by the branch length. That's an estimate of how far individuals are moving, and we can summarize that across an entire tree. We can also summarize that across specific regions of space or specific slices of time to learn about when reproductive isolation, prezygotic or premating reproductive isolation seems to be happening, and when it's not, if there are specific time frames where that's occurring, or spatial frames where that's occurring. The other thing, right, and this is from Sean's recent paper on speciation, Sean and many others on speciation, is that we can think about the geographic history of specific regions of the genome that might be involved in stuff that's interesting with respect to the speciation process. So it'd be fun to think about this for things like the MC1R locus for the white sand, dark sand lizards, right? That's presumably a piece of the genome that is moving across space in a really different way from the rest of the genome. And we're excited about the application of this type of genealogy or georeference genealogy approach to learn about how different parts of the genome are moving across space through time. Also, obviously thinking about these BDMIs and how they're moving and whether or not it's even possible to identify those. So I think I might have gone a little bit over time. I want to end just by saying thank you all again for the invitation. This is really fun to get the chance to talk about speciation with other folks who are excited about it. Thank you to the folks in my lab, especially Mike, who led this work, and my funders from the NIH and NSF. And with that, I'm happy to take a few questions if there's time.
Gideon, thanks so much. Really exciting and stimulating talk, I have to say. So we already have a question in the chat, which we'll start off with. So Conrad Loos asks, many tree reconstruction methods slash heuristics implicitly assume a lack of population structure. Is this assumption a worry if we interrogate the inferred tree sequence to learn about spatial structure slash history?
Yeah, that's a great question. I'm not going to have a satisfying answer for you. I think, most of these methods are inferring something related to either the coalescence rate or the inverse coalescence rate if they're doing demographic inference. The coalescence rate just is what it is, right? It's a function of the amount of sequence divergence between a pair or a set of sampled sequences. And so I don't think that it's necessary that be affected by like Wright-Fisher assumptions or something like that. And in general, I would say I would expect a lot of the tree reconstruction methods themselves to be more robust than things that try to infer demography from trees, which can be really stymied by structure. Or there was just a paper that came out, like a preprint that came out a few days ago about the role of background selection in tweaking or biasing our estimates of population demography. But I wouldn't say that I'm super concerned about the role of population structure in biasing the inference of the tree itself or the set of local trees itself.
Yeah, so just to follow up there, I guess. So if you do this analysis with something deterministic like TS-infer, and then you compare those results to something like argweaver, which assumes that there is in fact, a coalescent process that's occurring in a single population. Have you tried any comparison like that to see if the results vary or?
No, so we've been following this super closely. Deborah Brandt had this paper comparing a bunch of different ARG inference or tree sequence inference approaches. And then since then, I think there's been at least one more that came on the scene from Yun Song's lab, Singer, which gives you this sort of Bayesian posterior distribution of possible topologies. And then I know that the TS date folks are doing a lot of work to try to update how the dating happens. So I think it's a field that's moving really fast. And I think we don't have a great sense. There aren't a great set of like benchmarked, here's how these different inference methods work on these different scenarios. But I would say, I guess I would say this, maybe this is hopelessly naive, but I think there's so many trees. And the really important thing is the topology of the tree, which I think is less likely to be affected than the node heights of the tree, right? Because the topology of the tree in our case is what's pulling the geographic, sort of the tree weighted centroid of locations is what's pulling us all of our ancestry back into Northeast Africa. I'm much more worried about the effect of patchy sampling or biased geographic sampling than I am about the impact of this type of structure on tree inference.
Okay, great. Any other questions? Okay, I think with that, we'll just thank Gideon and move on to the next talk. So yeah, thanks so much. Really exciting.
Thank you.
That was lovely, Gideon. And so we have a double header today from Michigan. So it's an honor to introduce our next speaker, Dr. Lacey Knowles, who's a professor of ecology and evolutionary biology at the University of Michigan. And Lacey is well known for her pioneering work on resolving sources of genealogical discordance, testing hypotheses in a phylogenetic framework, inferring species boundaries, and among many other topics. And I've always loved Lacey's work, and in particular, what I love is the seamless way that she blends and works with empirical data from some really fabulous natural systems, along with theory and simulations. So please join me in welcoming Lacey.
Well, thank you, Sonal, very much for a nice introduction here. Let me get my slides up. Are you seeing this?
Perfect.
Yeah, okay. Just need to move this bar that's right in the middle of my slide. Ooh. Okay. All right. Well, thanks so much for the invitation. It's a great honor to be able to talk to people interested in speciation. And as Sonal mentioned, my work really spans the realm from looking more at phylogeographic inference into the realm of species differences in a number of different contexts. And thanks to Gideon for giving this great introduction to the geography of speciation, geography of divergence. So I'm going to focus more on some empirical systems and different types of questions that we might address using genetic data in a spatially explicit framework. So I'm going to give an introduction to some general questions to introduce some methods that we use that are spatially explicit. But then I'm going to focus the talk mostly on cryptic diversification. How do we make inferences about species boundaries and what are the implications for quantifying biodiversity? And then I'll talk about some really recent work where knowledge about the speciation history actually informs conservation and restoration practices. So it's a little bit of a shift, but it's, I think, an interesting case where you have applied consequences of studying speciation between different groups of plants out west. So some of the methods I'll talk about, we call the IDDC methods, and these are methods which basically integrate coalescent models with information about the ecological distribution of habitat. So we can test different ideas about isolation by distance, for example, ideas about whether current divergence patterns reflect the current landscape, whether divergence patterns actually reflect shifts in the landscape or shifts in species dispersal over time as the landscape changes. So with these methods, we're basically start off with some type of distributional model, ecological niche model, which gives you a quantitative layout of habitat suitability scores across the landscape or across an area that you're interested in studying. And we take these suitability scores and translate them into a demographic model. So basically, if you have a high suitability, you have a high carrying capacity and a high probability of sending out migrants, which are shown in the parentheses. And then we save all this demic information to do a spatially explicit coalescent model. So instead of thinking of a coalescent model in just one population, the way people model just one tube, we're actually modeling the coalescent across space. So it keeps track of where your ancestors are across space and how far back in time they actually occur. Then we take all this information to do a lot of simulations to test different questions that you might be interested in. In the case of the lizards in the previous slide, we were really interested in what types of divergence processes characterize the current genetic structure that we see in these lizards. And so we take all those modeling approaches and run them through an ABC process, which I just outlined here that I'm not going to go into detail. But you're watching a movie of the demographic spread, which I guess is over already. But it basically allows us to do model comparison to learn about which divergence process is dominating the system. And in this case, it is this colonization by a dynamic ecological niche model. So dynamic meaning we model the distributions of species in the past using paleoclimatic data and how that shifts over time all the way to the present. And so to do this, when we do this, we can look at questions that you couldn't address without a spatially explicit method. And another example where we're talking about spatially explicit coalescence to test ideas about the geography of divergence, we're actually interested in understanding where ancestral populations might have occurred on space or in space. And so this is, again, a similar approach, and I'm not going to go into it, but it actually looks at allele frequency gradients using summary statistics to infer the geographic location of the ancestral population. So you're looking at a heat map that with the X is the highest likelihood for the geographic location. What's really interesting in this study is if people are interested in ancestral populations and where they might have been in space, the ecological niche models don't provide a lot of information because they're projected across the entire East Coast, basically. But when we use genetic data, we can hone in on this area. And what's really cool is this area was also the place of a macrofossil for this hickory species. So it actually sort of appended some ideas about where species lie during Pleistocene glacial cycles, because this location, this refugial, ancestral refugial population is very close to the ice sheet shown in blue. And we looked at other species, and this is not the case. So this is a ecologically adapted species to live in environments that would allow it to persist along the glacial areas or glacial ridge. In other cases, we use models to ask whether we can see species-specific effects on patterns of species divergence. So in this case, we're interested in thinking about how the interaction between climate change and microhabitat or ecology actually influences genetic divergence. And to do this, and we're comparing two different species, we have to, again, generate species-specific expectations for patterns of genetic variation. And we want those expectations to capture how individuals move across landscapes as a function of the underlying environment, but also when that environment changes. So in other words, we use the same approach, this IDDC approach, where we have two models that we fit the data to. We have one model where we're modeling how the habitat suitability changed over time using paleoclimatic data in the past, and as well as the present, and shifting those relative contributions over time. But we can characterize where the glaciers act as barriers. And so in this case, we use glacial moraine data to reconstruct glaciers specifically from external data. So the black areas are all places where montane glaciers persisted based on glacial moraines or geologic data. And then we have one where those are permeable. And so we can fit those two data and ask, or these two models, to the different species and ask the question, did glaciers actually act as barriers to species divergence, but only in the wetland species where the glaciers accumulate in the basically the valleys in the mountains, which displace the wetland species specialist. And then lastly, for this example, before we move on to delimitation, we can ask the question of what type of geographic configuration of in space do populations or the geographic configuration of populations in space that actually promote divergence. So this is a case in grasshoppers where previous work has shown that the timing of divergence dates to glacial periods between species. And so we're looking at how these species were distributed in the present, but all the past, and ask whether it's the current Sky Island population. So these red dots are basically the Sky Islands. They're high suitabilities in red. And the little stars, if you can see them, is where we sampled genetic data. And I should have mentioned, in all these data sets, we're looking at RAD-seq data that we generate genomically, except for the lizards, which was sequences from 24 genomic loci. But we can ask the question, is Sky Islands, just the fact of being on an island, sufficient to promote divergence, or does it have something to do with where you were in space in a refuge and how you colonize those islands? And I haven't talked about it, but when we do this, we actually estimate parameters of that demographic process. So we have estimates of carrying capacities, migration rates, ancestral population sizes, and mutation rate. And we compare using ABC again, which of the models have the highest likelihoods. And in this case, it was the colonization model. So the importance of being displaced and where you landed in space in the past was a strong determinant of patterns of species divergence. So I did that introduction just to emphasize all the diversity of ways we can use spatially explicit methods, and then talk about a place where we haven't done this. And so when it comes to understanding species boundaries using genetic data, or species delimitation is often known, we tend to focus on aspatial or non-spatial methods. And so I want to talk a little bit about this because this is an area that has been a subject of research. We're really concerned about, are we actually overestimating species diversity when you use these methods? So the predominant approach to inferring species boundaries and estimating how many species you might have is applications based on the multi-species coalescent. So the multi-species coalescent models basically commonly implemented in BPP, but they're the same across all the different models, no matter what type of statistical implementation you actually apply. You basically have different sets of species. Like in this case, you have a model with two species where A and B are combined. And under this case, we have a model where we're saying there's 3 species. And you compare the probability of the data under these two different under these models, and you actually can break them up further. So you can have a suite of models that maybe have 1, 2, 3, 4, how many ever species that you might be considering. And basically, you evaluate the fit of your genomic data to these models and come up with an inference about hypothesized species boundaries using this method. So this method makes a number of assumptions, and one of the important assumptions is that speciation is more or less instantaneous. So when you see genetic divergences dating to a particular time, speciation happens at that time. And we know that that's not the case. Speciation has a lag time. There's actually a duration to speciation. You have some type of initial population split, and then it takes some time for the formation of independent species, say through the acquisition of reproductive isolation or whatever species definition that you want to use. Most people would not or would say that there is a species duration. It could be long in some cases, it could be short in other cases, but nobody would probably argue that speciation is instantaneous. And one of the consequences of saying speciation is instantaneous, of course, is saying every genetic divergence is equated with a species boundary. So there's no intraspecific population structure, which is a very strong assumption that you're making when you apply multi-species coalescent. And this graph is just to show the consequence of making that assumption. So when you assume speciation is instantaneous and ignore the lag that takes time for speciation to happen, you end up overestimating the number of species using multi-species coalescent. So we did lots of simulations where we changed the rate of species formation, so basically different speciation duration times. And you can see that overestimate can be very high, and it gets even higher the longer it takes for speciation to occur. And that's really problematic, right? But that's because you're treating speciation as being instantaneous. You're not considering that there's some population divergences within species lineages. So if we just talk about lineages, the multi-species coalescent is a very powerful approach for detecting genetic divergences. But there's nothing in the multi-species coalescent that allows us to distinguish genetic divergences within species versus genetic divergences between species. And so you end up with situations like this, which I think are not where we want to go, where you have whole species being defined, in this case, Hemidactylus coalescens, based on the coalescent structure. And so basically the diagnosis is basically all the lineages with strong support of Bayesian under the Bayesian speciation delimitation model. And if we know that this strong assumption of speciation is instantaneous is actually really unrealistic, really draws into question, should we be actually defining new species using the multi-species coalescent? So where does spatially explicit coalescent models come into play? So basically, we can do the same type of approach, and this is work from a former postdoc, Arno, in our lab, where you're looking at a demographic model of colonization across Australia. So this is the same thing where we use environmental data to inform the demographic model of colonizing the whole space. And we do this with lots of different simulations. And so what you end up with is genetic structure that varies and will differ depending on what the underlying environment is and what the underlying carrying capacities and migration probabilities are. And what this allows us to do is to look at the robustness of the multi-species coalescent. So robustness meaning how accurate are those inferences when you have different things that we know affect genetic structure. So things like IBD within species, isolation by environment within species, geographic barriers in species, and just the simple process of stochastic population extinction generates genetic structure within species. So basically, we can ask the question, is the multi-species coalescent robust? And so what you're looking at here is simulated data where we've actually analyzed across the entire landscape samples from this area here and compared it to the genetic divergences from different samples throughout the entire space and ran it through BPP, the multi-species coalescent model. And what it shows us is where in space does the multi-species coalescent actually make an inference of 2 lineages. So keep in mind, this was a single population lineage process. So there was no speciation event. So what that is doing is it's identifying where in space does the multi-species coalescent actually call intraspecific divergences. So this is going to be really important too for thinking about how do we make samples or draw samples to make inferences and when it comes to species discovery to avoid the problem of confounding intraspecific divergences with intraspecific divergences. So in lots of studies, if you sample sparsely across space, you actually will, and there could be isolation by distance, but you only sample at very geographically distant spaces, the multi-species coalescent is going to infer multiple lineages when there's actually just one isolation by distance, one model. So using this approach, we can actually think about if we sample at a certain geographic distance, is that going to be accurate or inaccurate for our inferences? And then, of course, if we do this, it also allows us to avoid confounding the interspecific divergences with interspecific divergences. So we avoid the overestimation of population splits. So basically, under a single expansion model of isolation by distance informed by the environment, you can see there is this area where the model doesn't predict any kind of genetic structure. So in your empirical data, if you observe genetic structure between your sampled populations within this region, you actually reject the model of no speciation. And this is an area where you would hypothesize that some type of speciation process is occurring. So it allows you to hone in on geographic areas that have interesting genetic divergences that you wouldn't expect under a single model or a single species model. So I think I have enough time just to really quickly, and this will be fast. I just wanted to talk real quick about how can we use speciation research to inform applied applications in conservation and habitat restoration. And so in this case, we're looking at the divergence. Oh, I should mention, this is a collaborative research by a former student of mine who's now at the USGS, and a number of other collaborators working at USGS or University of Nevada. And we focus in on foundational species, and we're focused on foundational species because conservation efforts are very interested in how these plants basically become patchily distributed across the landscape, because as foundational species, they're really important to pollination systems and ecosystem function. And yet not a lot is known about their genetics. We have the hint that there's some type of hybridization in each of these three species because there's morphological intermediates that we can actually collect. So we collect the SNP data from across all those species, and we analyze it with different models of species divergence. And across all the three plant species, the strict isolation model is actually never inferred. Ada, I have a dog who, sorry, a dog who's not feeling well. It was on the couch. I was trying to get off. But what it shows is that there is some type of gene flow in the actual history of all three species. And what's interesting is despite gene flow, the parental species in all three cases, so this is the rabbitbush, the sagebrush, and the globe mallows, parental species remain genetically fairly distinct, with the hybrids obviously having a mixed ancestry of the two parentals. And they're not forming hybrid zones like clinal zones. They actually form mosaic hybrid zones where you actually have hybrids shown in yellow in all three of these plots being interspersed among the parental species. And then what's really interesting is we can actually look at the hybrids and do a RDA plot and look at the association between genotype and environment and show that hybrids are actually occupying intermediate environmental spaces. And this is true in the rabbitbrush, but it's also true in the sagebrush. You get the hybrids in an intermediate environmental space, and same with the globe mallows. And so that raises some really interesting questions about conservation and restoration, because if you're trying to avoid fragmented landscapes, it shows that hybrids shown here in this yellow from a projected occurrence using random forest models actually seam together or sort of act as sutures to stitch together landscapes between the parentals, which are shown in green, and purple in the case of the glow mallow here. So it's a case where we have an underappreciated role of hybridization in terms of a divergence history or evolutionary history where hybrids are forming, but they're not diluting the parental gene pools, but they can form an essential ecological function in terms of restoration. So typically you would avoid hybrids and restoration efforts, but by studying the speciation history, we can show that the hybrids actually can form or play an important role ecologically without jeopardizing evolutionarily the history of the parental species themselves. So with that, I think I left a couple minutes for questions. I know I talked pretty fast about different projects, but I'm happy to take questions.
All right, that was really fascinating. Thank you. It seems like some of the questions. Oh, we have somebody who raised their hand. Sean.
Yeah, thanks for a great talk there. I just wanted to clarify. So the issue about inter versus intraspecific speciation in the MSC, is this related to, or is this caused by the fact that the MSC is not spatially explicit, or has it got something to do with the specific colonization history of the organism?
So the MSC, basically, there's nothing in the coalescent models that would allow you to differentiate intraspecific and interspecific. So there is an inherent limitation because the assumption is all genetic divergences and under the MSC, and by genetic divergences, I should say significant genetic divergences, which are basically divergences that are too deep than what you would expect under a standard Wright-Fisher model.
Right.
You equate those with species differences. So the spatially explicit coalescent model was just a suggestion of how do we actually get expectations for how genetic divergence accumulates across space under realistic conditions. So we can use the environment to say, account for genetic divergence by isolation by environment or geographic barriers. And so by robustness, it's basically just saying what parts of geographic space are you likely to infer two species, like maybe you sampled at really distant places, you're going to infer two species on the multi-species coalescent, but by doing the spatially explicit model, you can see that the inference is incorrect. That's basically the exact what you would expect under a certain environmental condition of intraspecific divergences.
Yeah, great. So that was what I was kind of guessing. So I guess that leads me to the idea of like, would it be possible in your mind to have an MSC that actually does allow for geographic structure within species?
So there's nothing in the MSC that models intraspecific divergences versus interspecific divergences. So basically the multi-species coalescent, we just call it multi-species because we're applying it to multiple lineages. It's basically a coalescent model applied to multiple lineages. So there's nothing particularly magical about the multi-species coalescent. It wouldn't be any different than just asking whether the gene depth is deeper than you expect under a Wright-Fisher model. But since we're looking at multiple lineages, we do this in a tree format where the models actually have, one lineage, two lineage, three lineage, four, as up to as many lineages that you're interested in testing. But there is nothing in the multi-species coalescent that would allow you to differentiate between intraspecific and interspecific divergences.
Thanks.
Mm-hmm. Liz?
Yeah, thanks, Lacey. That was super interesting. So I'm curious about the role of varying environmental predictors and how that would impact the inferences that you would derive from these kind of spatially explicit approaches. So if it seems like a lot depends on what variables go into those environmental models. So I'm curious about how robust they are to varying kind of those input environmental predictors.
Yes. So, you could always do this under a diffusion model without using any information about the environment, which is making a lot of strong assumptions. So obviously inferences about the environment using ecological niche models have been criticized for not being particularly robust in some cases. All you can do is apply all the standard techniques to ecological niche models to try to make sure that you're not overfitting the model and that it's giving you good projections of habitat suitability. So there basically are a lot of assumptions built into this by using the environment, but I would argue that it's not any stronger of an assumption by saying environment doesn't matter. So if you do a diffusion process, you're saying environment has no impact on dispersal capabilities or even local carrying capacities. And I would argue that you're better off, even if it's crude based on the ecological niche modeling, accounting for variation across space, because we know habitat suitability varies across space. So incorporating that information into your demographic model, I think, while it makes assumptions, it's less strict of an assumption as saying environment doesn't matter. But of course, you're right. If you do an ecological niche model and you overfit your model, or you didn't do any of the testing of it, and you're conditioning on a suitability that's way off, then your inference would be way off.
Thanks.
Mm-hmm.
Great. Maybe we can now open it up for a more general discussion. And perhaps I think I'd like to start by